In the coming presentation, I want to introduce a Walz instrument, the leaf state analyzer. At the same time, I want to talk about the processes that can be measured with it and, as well, the interrelationship between the measured parameters and photosynthesis. With the instrument, it is possible to obtain information on the optical properties of the leaf in the form of leaf angle, screening of different wavelength domains, and chlorophyll content. These optical properties directly and indirectly affect the photosynthetic activity. To link these properties to photosynthesis, the parameter Fv over Fm is determined. In this respect, the instrument complements PAM instruments, which purely measure photosynthetic activity without consideration of the optical environment. First, a short overview of the topics I will discuss. I will start with a look at the instrument, then a discussion of the way the instrument determines the chlorophyll content, the leaf angle determination, epidermal screening of UVA, UVB, blue and green light, the data output, and two special applications, the nitrogen content of leaves and fruit ripening. The measuring head of the instrument consists of a leaf clip with on the top side a detector and 5 LEDs emitting at 310, 360, 450, 530 and 630 nanometers, where the first 4 wavelengths emitting in the UVB, UVA, blue and green are used to determine the extent of screening of these 4 wavelengths and where the 630 nanometer LED is a reference wavelength. In the bottom half of the measuring head, 2 LEDs of 715 and 775 nanometer for the determination of the chlorophyll content are located. If the user wants to measure bulky objects like fruits, he or she can remove the bottom half of the measuring head by removing a screw and covering the electronics with a lid. With the lower part of the measuring head removed, it is no longer possible to determine the chlorophyll content. To put the leaf state analyzer in perspective, I decided to make a comparison between this instrument and the micro-PAM, which is used for monitoring. The micro-PAM monitors not only the photosynthetic activity, but also the ambient light intensity, leaf temperature and humidity. Using the photosynthetic activity, it determines the impact of a set of external physical conditions on a set of leaves and how these leaves cope with these physical conditions. The leaf state analyzer, on the other hand, measuring optical leaf properties, tells the user how the leaf copes with its light environment at that moment. If I think about this coping, the following keywords come to mind: avoidance, distribution, and screening, where avoidance and distribution can represent 2 aspects of the same phenomenon. The distribution of light can take 2 forms. Within a leaf, an extra layer of palisade parenchyma, allowing the distribution of light over an extra set of chloroplasts. A leaf angle other than horizontal, which allows its distribution of light over different leaf layers. Here, an ecophysiological example of a change in light distribution is given. Under certain high light conditions, newly developing leaves can form a second layer of palisade parenchyma. The number of cells does not change, but there is one division in the direction of the center of the leaf and not parallel to the leaf surface. An observation that has been made is that sun leaves are in general thicker than shade leaves. The figure of Yano and Terashima shows for sun leaves a more pronounced development of 2 layers of parenchyma cells, and the associated increase in chlorophyll per square centimeter can be detected with the leaf state analyzer. If we then look at the chlorophyll determination method, it is important to note that the chosen wavelength of 715 nanometers is much further to the far red than the 650 nanometers that is used in spot meters. As reference, 775 nanometers was chosen. At 775 nanometers, chlorophyll no longer absorbs. This approach follows the method proposed by Zerovic et al. In 2012 in Physiologia Plantarum. The spectra shown here illustrate the choice made. At 650 nanometers, the chlorophyll B peak is found, and this wavelength is on the rising slope of the chlorophyll A peak. Together, this gives a strong absorption signal, with one important disadvantage. Already at the relatively low chlorophyll contents, saturation occurs. At 715 nanometers, the absorption signal is much weaker, and as a consequence, saturation first occurs at much higher chlorophyll contents. The figure shows 3 calibration curves for C3 plants, Helianthus annuus, Nicotiana tabacum, and Triticum aestivum, and for the C4 plant Zea mais. The fit gives the calibration curve for all 3 C3 plants together, and another fit curve for the Zea mais data. The idea is that the C3 fit curve can be used as a proxy for the fit curve that could have been obtained for C3 plant species with which the user works. Choosing either the C3 or C4 fit curve in the settings, the software will turn the LSA data into nanomole chlorophyll per square centimeter and microgram chlorophyll per square centimeter using either the C3 fit curve or the Zea mais fit curve. No doubt, every user wants reproducible chlorophyll content data, and there one phenomenon has to be taken into account. Jan Naus and colleagues in Olomouc in the Czech Republic considered chloroplast movements and found up to 35% difference in the difference between 650 nanometers and 940 nanometers, inducing the 2 extreme chloroplast positions. To avoid unnecessary variability, it is a good idea to bring the chloroplast in the dark state position by dark acclimation. For this, green dark acclimation bags were developed. Dark acclimation is also necessary for the determination of correct FE over FM values. The second main function of the leaf state analyzer is the determination of the leaf angle relative to the sun. If you look around while walking, for example, in the garden, you may notice that many leaves are not oriented horizontally. This may have 2 reasons. It helps to distribute light over more leaf layers, and at the same time, this means that chloroplast in such a leaf will absorb less light. In the slide, 2 examples I saw in the gardens of our neighbors when I was working in Lullier in Canton Geneva. On the left-hand side, some leek plants with the central leaves being almost vertical, and on the right-hand side a palm-like composite leaf that as a whole is quite vertically oriented. If the leaf state analyzer measures the leaf under its natural angle, then the instrument determines the angle of incidence of the sunlight on the leaf, taking into account the position of the sun and the orientation of the leaf. To give an idea about the importance of light distribution over different layers of leaves, I took a model calculation from an article by Long and co-workers published in 2006 in Plant, Cell and Environment. The model calculation compares 2 cases, horizontal leaves versus leaves placed under an angle. Looking at the light response curves made for the horizontal case, plant X, the top leaf 1 is supersaturated, whereas the lower 2 leaves 2 and 3 are still far from saturation. If the leaves are oriented under an angle, plant Y, a better distribution of light is possible, with all three leaves being nearly saturated. The horizontal configuration yields overall a 30% lower photosynthetic activity. If we then look at the technical side of the leaf angle measurements, then you will see that it is by far the most sophisticated part of the leaf state analyzer. To sum it up, for the measurement of the leaf angle, the instrument needs a gyroscope, an accelerometer, and a magnetometer to determine the north-south axis, and for the position of the sun, a GPS receiver and a clock are needed. In the next slide, the two parts of the calculation are illustrated. The top figure shows that both leaf angle and azimuth, angle relative to the north, and the angle relative to the Earth's surface are determined. In the bottom figure it is shown that also for the Sun, the angle of the Sun relative to the Earth is determined as well as the azimuth. On the basis of these two calculations, the cosine of the angle of incidence is determined, on the basis of which the effective light intensity can be determined. For the leaf angle determination, the time zone has to be set up in the software that comes with the instrument. First a continent is selected and then a nearby city. For a relatively simple instrument like the leaf state analyzer, kinetic fluorescence measurements are not an option. The only parameter directly related to photosynthesis that is determined by the instrument is the FV over FM. For this measurement, the green LED is used as measuring light and the red LED is used for the saturation pulse. Finally, the last main function of the instrument, the determination of the extent of epidermal screening at 4 different wavelengths: 310, 365, 450 and 530 nanometers. For each of these wavelength domains, molecules can be found in the epidermis of leaves that can absorb these wavelengths. Hydroxycinnamic acids can screen UVB light, flavonoids can screen UVA light, carotenoids can screen blue light, and anthocyanins can screen green light. Screening of UV light is important because UV light can destroy the manganese cluster of photosynth 2, and it can cause damage to DNA. For this reason, it is important for plants to reduce UV levels strongly by epidermal screening. The screening of blue and green light has been suggested to be more related to the reduction of excitation pressure and thereby to reduce the risk of photoinhibition. The fluorescence-based screening method was developed by Wolfgang Bilger and Ulrich Schreiber and was published in Physiologia Plantarum in 1997. The cartoon in this slide tries to illustrate the principle of the screening method. It is maybe not widely known, but chlorophyll molecules can be excited by UVB and UVA light, and this leads subsequently to the emission of fluorescence. If less UVB or UVA light reaches the chloroplast, less fluorescence will be emitted. The emitted fluorescence is proportional to the light intensity that excites the chlorophyll molecules. This, however, is only true if the excitation light is not actinic. In the original methods, blue-green light was used as reference, but since we know now that blue and green light can also be screened, in the leaf state analyzer, red 630 nanometer light is used as reference, as suggested by Erhard Fündel and co-workers in a paper published in Photosynthesis Research in 2007. However, as will be explained below, the determination of screening requires a more complex calculation. Before continuing with the technical side of the screening measurements, let's first have a look at the processes for which screening may be important. As mentioned already above, UVB and UVA screening are important for the protection of DNA and the manganese cluster of photosystem 2. The screening of blue and green light may reduce photoinhibition under high light conditions. That screening of blue and green light may have an impact on photosynthetic activity was shown in 2017 in Planta by Nichelman and Bilger, who observed for red Berberis thunbergii plants that a 40% screening of incident light by anthocyanins led to a 40% reduction of light-limited photosynthesis. In some shade plants, screening pigments are found on the bottom side of the leaves. Fernandez Marin and co-workers suggested in New Phytologist in 2015 that this may be a strategy. By reducing the transmission of light, it becomes darker underneath such plants, making the growth of competitors more difficult. Another issue is the transient presence of anthocyanins. Chalker Scott, in a review in Photochemistry and Photobiology in 1999, notes that anthocyanins are often transiently present in juvenile and senescent tissue, and that anthocyanins can also be induced under certain stress conditions. In juvenile tissue, the photosynthetic apparatus has often not yet reached maturity, and that means as well that it is not yet 100% functional. Under these conditions, the presence of anthocyanins will reduce the excitation pressure, protecting the immature photosynthetic apparatuses. The same is true in senescing tissue, where the photosynthetic apparatuses have to be degraded without causing oxidative damage, and where anthocyanins help by reducing the excitation pressure. Under cold stress conditions, the photosynthetic biochemical reactions slow down, whereas the photochemical reactions can occur with the same efficiency. There again, the presence of anthocyanins reduces the excitation pressure and thereby the risk of cold-induced damage. The leaf state analyzer is almost a push-button instrument. The calculation of the screening is done by the instrument. However, it may be worthwhile to have some knowledge of the applied method. The epidermis of some plant species can be removed relatively easily. From 3 of these plants, the epidermis was removed and the fluorescence emission at the 4 screening wavelengths and the 630 nanometer reference wavelength was determined. Under these conditions, there is no epidermal screening. Under these conditions, it is possible to obtain the efficiency of fluorescence emission at the 4 screening wavelengths compared to the reference wavelength. This is called the mesophyll reference. This reference is instrument dependent, and to make it instrument independent, the same ratios are determined for a fluorescence standard. Dividing the mesophyll reference by the ratios determined for the fluorescence standard gives an instrument independent reference value, RF lambda. The next step is to turn the instrument independent RF lambda value into a mesophyll reference, MR lambda, of the instrument of the user. To do that, RF lambda is multiplied by I lambda divided by I 630 nanometers, determined with the fluorescence standard. And finally, to determine the screening by a leaf, the I lambda divided by I 630 nanometers of the sample is determined for the 4 screening wavelengths and divided by MR lambda of the device. The photo shows on the left side free-lying parenchyma of Helotelefium, and on the right side the grayish-looking removed epidermis. In a perfect world, it would be possible to determine the mesophyll reference for each species of interest by removing its epidermis. However, in practice, there are only a few plants of which the epidermis can be removed without problems. For the LSA-2050, mesophyll references were determined for Helotelefium, upper and lower side of the leaf, a tulipa species, and Kalanchoe degramontiana. The slide shows the mesophyll reference factors for the different plant species at the different wavelengths, values the instrument uses for its calculations. The software that can be installed on a PC or laptop has only a few functions. The opening of the software gives 3 items: the serial number of the instrument, a button that gives access to windows that allow the setting of the time zone, and a list of measurement files that can be transferred from the leaf state analyzer to the computer. To transfer a measurement, click on that file and click Yes when the program asks if it should save that file as an Excel file. This slide shows what such an Excel file could look like. On the left-hand side of the sheet, the FVFM value is indicated, then the mesophyll reference used, then the measured screening at the 4 different wavelengths. In the case of the flavonoid and anthocyanin screening wavelengths, these values are turned into absorption values, A-flav and A-ant. Then 2 columns with chlorophyll content determined as nanomole and microgram per square centimeter. Then the parameter MBI, which has been introduced as a measure for the nitrogen content of the leaf, and finally a parameter related to the leaf angle of incidence. As shown in the next slides, there are data needed for the calculation of these parameters, which are made invisible, but which are there. There are 2 ways in which the hidden information can be made visible. The arrow points at numbers 1 and 2 found on the upper left side of the sheet. By clicking on 1, the spreadsheet width is minimized, and by clicking on 2, the spreadsheet width is maximized. The alternative approach is to make use of the 3 pluses found above 3 different sections of the spreadsheet. In the slide, the first plus was clicked, making additional fluorescence and screening values visible. Here, the second plus is clicked, making 4 additional columns related to the calculation of the chlorophyll content of the leaf visible. And finally, clicking the third plus, columns with data related to the calculation of the angle of incidence are made visible. This makes the column leaf slope visible, which gives the angle between the leaf and the earth. Measuring the top side of the leaf, this gives values between 0 and 90 degrees. If the instrument is turned around to measure the bottom side of the leaf, it gives values between 90 and 180 degrees. It is possible that the spreadsheet contains negative A-flav and A-ant values. Here I was measuring top and bottom sides of leaves without changing the mesophyll reference, and as the spreadsheet shows, that is not a good idea. If the user gets such negative values, it is a good idea to try another mesophyll reference. Zoran Zerovic and colleagues introduced in 2012 a parameter that allowed the estimation of the nitrogen content of leaves. When considering the LSA-2050, a change in the nitrogen content has 2 effects. A lower nitrogen availability leads to a lower chlorophyll content. Chlorophyll molecules have a high nitrogen content. On the other hand, plants tend to increase their flavonoid levels under those circumstances. The NBI considers both aspects and gives better results than a parameter only considering chlorophyll content. Epidermal screening measurements can also be used to monitor and determine the ripening of several fruits like olives and grapes, but also apple peel, where an increase of the epidermal anthocyanin levels occurs. Agati et al. Argued in 2007 in Journal for Agricultural and Food Chemistry for grapes that on the basis of the anthocyanin content, it is easier to determine the perfect harvest moment than on the basis of the sugar content of the grapes. To finish this presentation, 2 examples of measurement sets. First, measurements with the red Berberis thunbergii. For this plant, based on the color of the leaves, you expect high screening levels of blue and green incident light. Two variables were considered: shade leaves versus sun-exposed leaves, and the upper sides versus the bottom sides of the leaves. With respect to UVB and UVA, it is quite clear that sun-exposed leaves contain more screening pigments than the shade leaves for both sides of the leaf. With respect to blue and green light, the top sides of the sun-exposed leaves contain more screening pigments. Effects of the different configurations on FV over FM and chlorophyll content, on the other hand, are limited. The second example compares common beans grown either in the field or in the greenhouse, where measurements of the top and bottom sides of the leaf form again the second variable. The common bean is green, and as expected, there is almost no screening of green light, and also little screening of blue light, although the percentage screening is somewhat higher in the field than in the greenhouse. Field-grown plants show a stronger screening of UVB and UVA than greenhouse-grown plants, and especially with respect to UVA, there is a considerable difference between the top and the bottom sides of the leaves. The data further indicate that the FV over FM data of plants grown in the field are higher than those of plants grown in the greenhouse. An explanation for this could, for example, be a difference in the PS2 to PS1 ratio. Also, the chlorophyll content is somewhat lower in the greenhouse than in the field. I hope that with the description of the instrument and the different examples of applications, potential users may get a feeling for the research possibilities the instrument offers. Thank you for your attention.